Hello, everybody. Welcome to another Pint of Science, the Netherlands. Today we have our a third talk of the week. So it's about our second brain. And when you get on your gut nerves, today we are going to hear all about it. Today we are with me, Margarida. I'm a PhD student from the University Medical Center of Utrecht, and with Linda.
Yes. Hi, everyone, and welcome to this event. So my name is Linda. I'm a postdoctoral researcher at the Institute of Data Science, which is part of Maastricht University. And I'm also one of the city coordinators of Pint of Science Maastricht. So we're here today with two very special guests from Maastricht University presenting about their research. So I would like to welcome Veerle Melotte and Nathalie Vaes. Veerle is an associate professor and Nathalie is a postdoctoral researcher. Both working at the School of Oncology and Developmental Biology. Veerle is next to the also affiliated with Erasmus MC Rotterdam, and they both work on investigating the connection between the enteric nervous system and colorectal cancer. Veerle is the winner of several personal grants and has also developed an FDA-approved biomarker for the early detection of colorectal cancer. And Nathalie is currently working within Veerle's lab to investigate the relationship between the second brain and colorectal cancer in zebrafish. So with that, I would like to give the floor now to Veerle and Nathalie, because I think they have a very interesting talk lined up. So the floor is yours, ladies.
Okay, thank you very much. So thank all at home or for joining. Unfortunately, we're not in a pub as it's supposed to be, but we're all at home. But I hope you sit and relax and enjoy a beer at the moment. We have one. So
cheers, by the way.
Cheers. 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 So thanks for the organizers and Linda for giving us the opportunity to present our work here today. For those at home, please don't drink too much because you have to stay focused, because we have a little quiz at the end, and at the end you can win a Pint of Science. Don't have an example. It's that one. Yes. There's really a prize. So if you really want to go for the prize, stay focused and stay sober. But we're not here to talk about beer, although I also have to say, Nathalie and I, we are both Belgian, and we are in Belgium, so we are beer lovers. We will talk about the second brain today. So I think you all know our first brain. That's familiar for everyone, and we have here an example how it looks like. You have to be careful that it doesn't fall apart. So this is how it looks like. I hope you all see it at home. So there's the second brain, and it's hidden behind your skull. And we don't all know that we also have a second brain within our belly. But I think you all know at home that, for example, you already had once butterflies when you were in love and when you were really exciting, or that you, for example, had pain in your body or in your belly when you were stressed, or when you were not feeling comfortable. And this is all related to the second brain, because in your belly there is a huge second nervous system. Now, going back to the structure, you see here how the first brain looks like, and I'll also just show you. And also, if you take it out of the skull, it still is a very nice structured organ. This is totally different for our intestine. If you see it here, when it's outside of the body, it's quite a mess, and it doesn't look very structured. It does look very chaotic. Although, if we go more in depth to the structure or to the organ, we also see that it's very structured. So you see here the different layers of the gut. So normally, it's a ring structure, of course, and the white space in between, that's where the food processes. And you see here that there are also nice structures. So you have a mucosal layer, submucosal layer, and different muscle layers. So the second brain, it's situated between these different layers, which you see here in yellow. And I would like to go to here the other figure. It's again, a part of the intestine. Here it's again where the food passes by, and you see again the different layers. And there are in yellow here 2 thin layers, so-called submucosal plexus and mesenteric plexus. And these two layers are the second brain. And these thin layers, they go from the stomach to the anus. And altogether, they're very big and they resemble the second brain. And here you see, if you would peel off the different layers from each other, you see that the cells are nicely connected and yeah, that they really resemble a brain. Now, there are different cells within the second brain. There are two major cell types, the glia cells, which you see here, and also the neurons, and they are in a ratio 6:1. So you have more glia cells compared to neurons. And both cell types are situated in the so-called ganglia, which you see here, again, in the mesenteric plexus and in the submucosal plexus. Now, to come with a little bit more facts to compare the second brain and the first brain. So within the first brain, so our brain underneath the skull, you have around 170 billion neurons or glia cells, which is a lot. And together with the spinal cord, the brain and the spinal cord, they are the central nervous system. I think you all heard of the central nervous system. So within the gut wall, you also have an enteric nervous system, and these consist of more than 200 million neurons and glia cells, and it's the size of a cat brain if you take all these glia cells and neurons together. So it's quite huge, and that's also a reason why it's called the second brain. Okay, so I already told you at the outside, the first brain looks much more structured compared to the second brain. And this is also when you would look specific at these neurons. So within the brain, it's a really nice organized layer of neuronal cells. And this is not the case in the gut, because in the intestine, there's a random distribution of neurons. And that you see in here, in the next slide. So you see here at the left, I think for all of you, it's a slide of a brain tissue. And here we stain different, or we colored, so to say, different neuronal subtypes within the brain. And they each have a different color, as you appreciate here. And when we look at the second brain, you see that it's not so nicely structured compared to the first brain. And that's why, people are still, of course, investigating the second brain much more, because it's a new system. Much more is known about the brain, and we don't know yet why there are all these different subtypes and why there is so a different structure compared to the first brain. I think I have not the latest version of the presentation, but I will switch it when I'm done. So there is because it's so an important organ and it's quite big, you can see that it's important for various neuronal functions and gut functions. So that is because these neurons communicate, or they control different cell types within the gut. For example, these neurons, they communicate with muscle cells in the gut, they communicate with secretory cells in the gut, and they also communicate, for example, with blood cells. And by this communication, the second brain, these neurons, regulate all gut functions. And then, if you process food, so for example, if you have some bread in the morning, you will have features at the end of the day, and this is because, your second brain takes care of the digestion of this food. They also regulate the contraction of your gut, and they also take care of gut homeostasis. And as said, they do it via different messengers, different signaling molecules. And interesting to tell you, so it's not that it's, I have here again, and I hope you see it. So interesting to tell you is, so you have here a second brain in the gut, but it's not really separate from our first brain. So you see here the first brain, and you see here this white line going from the brain towards different organs, all organs of the body, but also our gut or our intestine, and that's how they communicate with each other and how they also regulate gut function. So the second brain doesn't always work on its own. But very interesting. Let's give this back. Interesting is what they found is that the second brain can also do its work very independently from the brain. So you see here in this movie, I will start a movie. So this is a piece of intestine taken out of the mice, and it's put it in an organ bath. And in the organ bath, there's a medium with different nutrients. And you see here that the white pellet, which you see here, passing by, it goes toward the end of the intestine. So the muscles still are controlling via the second brain the contractions of the pellet. So meaning that the second brain also really works independently from our first brain. So based on the fact that it's really big and also based on the fact that it can work independently, it's called again the second brain. And up till date, this is not known for any other organ.
Can I just ask a question about the previous video? Because I was wondering, when you have this, you see this tube, so this is just a gut without being connected to any other organ.
Yeah, yes, correct.
Yeah.
So you don't see any, because it's fixed at the start, at the end, to have it straight, but you don't see, so it's really only a piece of intestine indeed. And it's put in an organ bath, so you could also have it like just flowing around there, and then you also would see the contraction, but this looks more nicely, because here you see really the pellet movement. But it's really outside of the body, and it can work like this for around 2 more hours once it is taken out of the body. Yes.
Yeah, cool. And this, sorry, just very curious about it. Is this from a mouse gut?
Mouse. Yes,
yeah.
This is mouse.
Yeah.
So is that also the reason why you sometimes hear that there's still like gases inside of like corpses? Because the movement is still like ongoing after you pass away?
That's true. Yeah, that's indeed true.
Ah, okay, very interesting.
Yeah, so sorry, I'm always quick, but interrupt me when necessary. Yeah, so you can see it's quite an important organ, our second brain. And you can also imagine if there's dysfunction of the second brain, that many diseases occur. And I think they study already for a long time the second brain in the context of genetic diseases. And I here have one example, Hirschsprung disease. And this is when children are born with an underdeveloped second brain. So this is really a life-threatening disease, and these children also need, or their babies also need, an operation directly after birth. Because of its function in gut homeostasis and also contraction, you can also imagine if there's dysfunction of the second brain, you can also have gut inflammation or, for example, constipation, what many of us sometimes have. So this is also related to the second brain. So, and we in the lab, we found, and we really studied the context of the second brain in the context of colorectal cancer. So I already told you that the second brain communicates via secretion of messengers to different cell types of the gut. And in that way, it also communicates with tumor cells. So as you see here, so as I told you, the second brain is 2 plexuses, the submucosal plexus, the mesenteric plexus. It's far away from the tumor because the tumor develops in the inner circle. That's where this tumor starts developing. And so these neurons secrete messengers, and they communicate and they control this tumor growth. I will not go further into detail about this because it's quite complex, and also because of sake of time. But if you are really interested, you can also contact us afterwards, of course, after the talk, and then you can learn more about the second brain and colorectal cancer. This was my introduction. I don't know if it's possible if we can change PowerPoint. Can I stop sharing and share another one? Because
yes,
of course. The latest.
Yeah.
That should be.
Yeah, I think so. Just to take the opportunity to, because I think I forgot to mention, if you are you have questions, you are curious about something, just feel free to drop your question in the chat, the YouTube or Facebook, wherever platform you are using right now, because then we are going to answer your questions. So feel free if you, if whatever you would like to ask, just ask. In the meantime, I was also curious because you talk about neuron and glial cells. Is there a specific difference between these 2 cells and why they are important in the gut?
Yes, I will try to answer.
Shall I answer?
Yes.
Shall I try to answer? Well, there is indeed a big difference between the neurons and the glial cells, and the neurons were actually thought like being the most important ones, because everyone thought like a couple of years ago still that these were the only cells that could communicate with other cell types within the gut, and that the glia cells were only the cells that support the neurons, similar as what you have in the brain, that the microglia cells there support the neurons. But nowadays, people are more and more aware of the fact that the glia cells not only like support the neurons in a way, but that they also have their own functions, and that they also have an important function in regulating the intestinal motility and that sort of things. And Berend Boesmans in our group, he is also focused on these glia cells, and he also now wants to investigate if there is also a really important function for these glia cells in colorectal cancer or not.
Yeah, great, thanks. Yeah. We can share start sharing.
Yes.
Yeah.
So okay, so now that you have a lot of knowledge about the second brain, I want to dive into the lab and show you a little bit of how we study the second brain in our lab. Let's see. Go. Yeah, so of course, if you have the human gut, it's about 7 or 8 meters long. So you can imagine, if we want to study the human gut, we need to have short pieces, which we can study here, and that is usually obtained via routine biopsy, and then we can get them here on a glass slide. But if we, for example, what we also do in the lab is use mouse tissues, and what we then do is we have a mouse gut, which is approximately 30 to 40 centimeters of length, and then we can visualize the entire mouse gut on one glass slide, and this is what you see here, because here we have different pieces of the intestine, which are formed into a Swiss roll. And maybe the Swiss roll rings a bell with you, and this is very possible because Swiss roll is actually also a very delicious cake. So hopefully next time when you eat a Swiss roll, I hope you will think of this talk and remember us again. And next to these different tissues that we use, we also have a lot of cell culture going on into the lab. And we also use different mouse and zebrafish models so that we can try to figure out how human diseases actually work together. So if we then have tissues, we of course need to dig in deeper into these tissues and really see what is going on in there. And therefore we have a lot of visualization methods. And before we could actually see some things, we of course need to collect this tissue. So this is what we represent here. We first collect the tissue. Here, this is from a mouse. And on this slide, you see that there is a lot of preparation and processing of this tissue before we can further look into the tissue. After processing it, the gut and putting it into a Swiss roll, we put it in some sort of wax, which is called paraffin. And to finally get into the glass slide, as what you saw on the previous slide, we have to put this paraffin block onto a microtome so that we can cut these sections into really thin paraffin slices. And this is what you then see here. Here you get a glass slide with a thin paraffin slide. These slides can then be stained with different kind of methods, and they can be visualized under the microscope. And if we use these sorts of tissues, we will mainly make use of a technique which is called immunohistochemistry. And I will not get you bored with all the details that go around this technique. But the main message, what you have to keep in mind is that when you do immunohistochemistry, you will finally get an end product, which is a brown color when your cell of interest is there or not. So this is what you can see here. When you stain the colon from a mouse and from human, you can see here that there is nice staining here with a marker that stains the enteric nervous system. So if you remember what Veerle said before, you have this submucosal plexus up here, and you have the myenteric plexus of the second brain, which is located in here. But okay, so then we know that there is a staining in the enteric nervous system, but with this technique, we cannot distinguish if the neuron cells or the glia cells are stained, or if both of the cell types are stained. So if we really want to visualize one cell type or the other cell type, we really have to go into another technique. And therefore, we also collect the tissue in a different way. So what we here do is, again, after collection of the tissue, we will again cut open the intestine here. So as you can see here, we will then pin it flat so that you can start peeling off the inner layers of the gut, as Veerle explained. So what you see in this picture is that we started peeling off the mucosal and the submucosal layer. And when that process is done, we will turn the tissue upside down so that you will have the outer layer of the gut on top. And then we will again peel that outer layer off so that only the myenteric plexus will remain. This is what you see in this picture. And if you put this on a microscope after you put some nice fluorescent colors on it, you will get an image that will look like this. And before we get to this image, we of course need another technique, and it's essentially the same as immunohistochemistry as explained before. However, here you will not get a brown product in the end, but you're able to label your cells of interest in a yellow, in a green, in a red color, depending on which cell type you really want to visualize. And this is an example of how the enteric nervous system or the second brain looks like if you really stain for different cells, is what you can see here. And then you can also see if the different cells that are stained, if they overlap with each other or not, and if they're connected one way or another. And then finally, we also have another visualization method, and this will be explained by our colleagues in the lab. So let's hope this movie will work.
So here we have a frozen block with brain tissue inside, and we are gonna cut it now to use it later on for some stainings. And you can see the tissue. So I am doing an antibody staining, and with this antibody staining, we are gonna visualize the neurons and the glial cells of the gut. And then with a microscope, we are able to visualize this. So I'm now pipetting the antibody solution onto the slides. And we're gonna incubate it like this, wash it later, and then we're going to the microscope. And then after the staining, we can see through the microscope the tissue and the fluorescence.
So this video actually kind of wraps up the things that I already mentioned before. So if you maybe have questions about the video or about any of the other things, you of course can always interrupt us and ask us the questions. Yeah, so next to that, I already explained that we also have different models that we use in the lab. So one of the main cell culture models that we are actually using is that we are trying to make mini guts in our lab. So those are called intestinal organoids. And for this, we will again isolate the intestine. We will process this in a slightly different way as what you saw before for the plexus preparation and stuff. So actually, we will make sure that the whole intestine will be digested and washed a lot. So there are a lot of steps involved. But the end product is that you will get individual cells from the intestinal tract. And these will then be plated in some sort of matrix shell, which is like a viscose substance where the cells can be put into, and then they are able to grow into a 3D structure. So if you visualize these under a microscope after 1 day, you will only get these small kind of balls, you would say, but after a couple of days they really start to grow and they really start to make all kinds of weird shapes. So it's really nice to look at them under the microscope. And of course there is always a lot of imagination going on when you see these organoid structures forming. So I guess you will maybe also see a plane in here, or maybe some kind of a weird donkey with a really bad hair day or something. So it is always really fun to look at these organoids under the microscope. But of course, we are also very, very, very interested in our second brain. So we also need a technique to isolate the second brain. And this will also be explained by our colleagues in the lab.
the small intestine to make primary cell cultures. So first we flushed it, and then we are going to remove the fat under the microscope.
So at this point, we have a little bit of the small intestine. It's nice and moist. And what we want to do is we want to use the combination of 2 tweezers to place it on top of this pipette, like so. We get a cotton bud, like your classic cotton bud. Keep it a little damp. And what you do is we're first going to peel the outside off. So we'll make a cut the fat off. There's a little, let's say, opening, and we're going to use the cotton bud to drag downwards and to the side to take off the peel. And this peel is essentially the longitudinal muscle with the myenteric plexus. And this myenteric plexus, now, if only our cotton buds are a different color, then you could really see. But the myenteric plexus has the cells in it, which we're going to create the cultures. And there's a little itty bitty one that you see there. And then what we do then is, after we've done this quite a few times, we will take it to the cell culture. We will take it to the cell culture lab, where we will continue with the digestion and make the primary cultures.
So, as Veerle said before, our second brain is able to communicate with the different cell types that are in the gut. So in our lab, we are focused on colorectal cancer. So we want to investigate how these cells can communicate with the intestinal cell type. So therefore, we made some sort of a co-culture model where we have both the second brain and our organoids together. So here is a schematic overview of that. So at the bottom of this well, we would culture our mini-guts or organoids, and we would also culture the second brain. And what we then did is actually use the medium that wherein the second brain was grown. We would put this medium via this transwell membrane on top of the organoids, and with a microscope, we would visualize if this medium could actually influence the growth of the organoids, if the organoids would grow faster, if they would grow slower, and we could just visualize that using a microscope, looking at the organoid growth in a couple of days. So usually it's about 5 or 10 days.
Can I ask something here?
Yes,
on the previous scheme. So
yeah.
These neuronal cells,
Yeah.
You isolate them from, it's the same procedure as before, so from the gut, right? From the intestine? Yes. Okay. And then you co-culture, well, yeah, you culture together, you grow together these organoids, these mini organs of the gut with this medium. But why do they, I don't know, do they release something? Why not do them together immediately, like indirect communication rather than this kind of communication? I don't know if I...
No, that's a very, very good question. So at first we actually wanted to try and put the second brain and the organoids together, but it is a very difficult procedure, and people are still nowadays trying to figure out all the right media so that both the second brain and the organoids can grow together in one dish. So therefore, we chose to use the medium that was derived from these neuronal and glial cells. And it's actually known that neurons and glial cells can secrete different sorts of factors. So therefore, we chose to use the medium, which was put on top of the organoids via this transwell membrane, so that not all the nutrients and all the medium would immediately go towards the organoids, but only like the small signaling messengers would really go through this membrane towards the organoids. So in that way, we know that there are like signaling molecules that were released from the second brain and communicate to the organoids and can enhance or inhibit the organoid growth.
Okay, so does that mean that, for instance, a neuron cell that's on the side of the intestine can influence like this part of the intestine of, I don't know, exactly on meters of distance?
Yeah.
So yeah, so they influence each other, like they can just communicate like in distant.
Yes. Yeah.
Yeah. Well, that's cool to know.
We think so.
Yeah, I think interesting to add here is that there's also indeed secreted factors, but for example, in brain tumors, they show also that there is a synaptic activity between these neurons and these tumor cells, but then they have to be in more close contact. And we're still trying to investigate if that also happens in colorectal cancer. So that we don't know yet. So that's something we are aiming for to find out.
Okay, and then I will continue with our final model, which is the Zebrafish model, and we can also do that in Maastricht University because we also have a Zebrafish facility. And here I will show you a really short movie of the Zebrafish facility. So here you see how the adult fish are housed in the Zebrafish facility, and this is how you look at the zebrafish under the microscope. So these are really the tiny embryonic zebrafish that are only a few days old. So we actually think that the zebrafish is a pretty cool model to use, and it has some advantages. You can get a lot of zebrafish at once. So if you would breed zebrafish, you will get like 50 eggs from one breeding. And in the very early life stages, the zebrafish are also transparent. So as you can see in this picture. And because of their transparency, it is also very easy to follow different cell types in time. So what you see here, this is of course our main interest, because here you see that the enteric nervous system is labeled in green. And here on the, is it the right or the left for you guys? I'm really bad with those things. But here you see that if you have different labels on different cell types, so the red and the green, you can image the different cell types in one clear picture. And we can also do different motility assays to assess if the second brain is still fully functioning in the zebrafish model. So here you can see a movie of the zebrafish intestinal motility. And if you look carefully, you can see that there is movement in here and that the fecal pellet will drop in here. So I hope that I have showed you that our lab work is kind of like rocket science, or at least we think it's rocket science. Maybe you think it's more of a cooking lesson or so, but we really enjoy our rocket science every day. And now I give the floor back to Veerle for the final part of our talk.
Okay, so I hope you're still awake and still have brain cells left enough for these, only a few slides left, so don't worry. Now maybe you are brain dead, but then maybe because of the alcohol you're in a good mood. Either way is good for me, because I think you also heard already a little bit that the food can also regulate or control how you feel, and this is really a rising interest in the field. So this is the gut-brain axis studies, and here they focus on different interaction between the gut and the brain. For example, they study that when you eat well, that you are more focused, that you're happier, that you're smarter. And maybe you also found already many books that they're now everywhere available, focusing on this gut-brain axis. I'm really sorry, they're all in Dutch here, but I can translate. So it's like they say the intestine thinks along, or the food machine, the charm of your intestine, the brain of your belly, smart and happy, because of healthy intestine. So it's all related to this gut-brain axis, and yeah, much research is done in the field. However, what I also find very interesting is that now it's also known that the second brain also is really involved in central nervous system, so first brain diseases, as for example, you all know Alzheimer, Parkinson, and yeah, depression. So some examples for Parkinson patients, they also very often have digestive diseases or complaints, for example, constipation. So there's really also there a relationship. And what I find really interesting is, for example, in Parkinson patients, they have like a kind of protein deposit within their neurons. You see that in Parkinson patients, neurons in the first brain. And researchers found that you also see already these protein deposits within the intestine, so in the second brain. So it's very interesting if people maybe could diagnose or predict all Parkinson, for example, by looking at the gut. And I think it's also very interesting, of course, because you can imagine it's much harder to take some brain tissue because it's behind the skull, but also because every neuron within your head has a function in your body, but this is totally different for your gut. So there you can more easily take a biopsy, take a piece out to investigate the second brain. So I think there's a lot of music in this field and in this research. So looking forward to what is coming. And then, my final slide, or our final slide of the day, we are Belgian, we start with beer, we have to end with chocolate. And also, I think there's a very interesting feature. Here's some chocolate. Not Belgium always have chocolate, and Nathalie always has chocolate with her, so she had it already in her purse. So there's a very nice fact, also related to the gut-brain axis. So when you eat chocolate, I think you all know that you get happy from it, and that's because serotonin is produced. And interesting to say is that only 5% of the serotonin is produced in the brain, in the first brain, but 95% is produced in the second brain. How this relates and how if the serotonin which is produced in the gut can really go to the brain, I think it's all still a question mark. But here again, there are connections between the gut-brain axis and food. Okay, so with this, we want to end up this talk, but not with saying thanks to the whole group. I think we really have a huge enthusiastic group of people where we're working with. I think you saw it because they were all involved here and especially made all these videos for Pint of Science. It's not that we had them already available in the lab, so I think that's really nice. And our background is more colorectal cancer, and like Nathalie already told us, Veerle Melotte, he joined the group because he's an expert in the second brain, and that's how we now work already a very long time on the second brain with all of us. And the funders. So thank you, and we are happy to take every question. Or maybe first a quiz, I don't know.
As you wish. Well, thank you for your talk. Super interesting, but I guess first we'll go to our quiz, right? Yeah. Maybe, well, while the quiz is not popping up, maybe we can answer a question. There's, I think, already some questions popping up from YouTube. Linda, do you want to go ahead?
So someone is asking if there are other model organisms to study the gut, and when you get a major breakthrough, I wish you many, how do you test if it's applicable to human as well? That's a very good question, indeed.
Shall I answer? So maybe, there are other models indeed. So we also, and Nathalie showed you the fish and the cell cultures, but we also look, for example, in mice and mouse, and there are also people who are looking really in, different other models. So the question is indeed, is it transferable to humans? So I know that nowadays they can also isolate already glia cells and make these cultures from human tissue. So there you could also use, it's not what we are doing yet in the lab, and I think it's also harder to do, but you can also make cultures from human tissues. And of course, when we look at proteins which are important for, which we think are important, for example, and the link between second brain and colorectal cancer, we always also look in stainings of, we also color these human tissues. So we also check always first what is going on in the human. Are they also expressed in the human second brain, not only in the my second brain? So this is how we translate. And I also have many breakthroughs. Thanks for the wishes.
Well, thank you for your answer. I think it was quite clear. I think we can share now the quiz. Get your phones ready because you can join, via the phone. Yeah, you can, if you access, it says join at www.kahoot.it. So there's actually an app. I don't know if you happens that you have an app. But if you access, then it will ask you for the game pin, and then you can type these numbers. And once you join, it starts to pop up the players, and then we can play. I guess time. I think time for another question. Maybe we have another one.
I also have a question prepared. Indeed.
Well, I was just wondering. Yeah, well, okay, maybe do this one, and then the next you ask Linda. Sure. Sorry.
So Thijs says, thanks for the nice talks. I was curious to know how what you study differs from the method of investigating gut flora via microbial screening. Is that a different research area?
Well, yeah, the whole microbial research is actually a whole new research area, as what that you already mentioned. So there is a lot of interest in this regard right now, and our methods differ because you really have to make sure that your gut tissues are really processed in a proper manner. So when we do it, we always have to digest our tissues and make sure that we will get our cell types out of there. But if you really want to study the microbiome, then you will have to collect feces, or you would have to screen in the feces using different sorts of techniques to see which gut microbiota are actually in there. So it's indeed, actually a lot different than what we do here with our tissues. You could do it in the same study, of course, because it's easy, of course, to collect the feces from a mouse, and then after you have finished your study, also collect the intestines for the other methods that we are using at this moment. But it's indeed a complete novel research area.
Okay, thank you. I think there are 7 people who joined the quiz now, so I'm not sure if we should start or wait a bit longer.
Yes, well, we start. Get ready.
Yay.
We can win a class, Pint of Science class. Well, I will read the first question. So, is your second brain as big as the brain of? So we have 4 options. Can be dog, cat, frog, or rabbit. So here goes your best bets. We don't know. I also don't know. Whoa. Okay. 3 people make it right. That's nice.
Oh,
ooh, louder.
Next question. So which 2 cell types define your second brain? So you can choose from macrophages and neurons, muscle cells and glial cells, neurons and muscle cells, or glial cells and neurons. I think that's a difficult one.
Think so too. I particularly like macrophages, as I'm actually working with them. So macrophages, who doesn't know, it's an immune cell type that's actually all around our body. So if you have a cut on your finger, they will be there to protect you. Very important cells.
But maybe not a likely answer then.
They're everywhere, huh? They're everywhere.
We try to stay away from macrophages because it's a Chinese sometimes, like so many times.
Yeah, true. I don't know the pain.
There's also a lot of communication between the macrophages and the neurons in the gut. So
Yeah, very interesting cells. Yeah. Okay, cool. Okay, which machine is used to cut slides? Oh, here we go. So microtome, microslicer, microscope, or microphone. This is a tricky one.
yeah, we only mentioned it. It was not on the slides.
Yeah.
No. Linda, do you know?
I'm doubting between two answers.
Well,
Yeah, so I thought microslicer or microtome. But,
that was a tricky one.
I
Let's see
don't
who's
think it
leading.
exists. It was
We think we just made the word up for the quiz.
oh, okay.
Well
it does
then.
make sense, but okay. So next question: give the name of the two different plexuses of the second brain. It's a multiple choice. So is it either submucosal plexus, myenteric plexus, or both, or you can tell us?
Well, it's there are tricky names. Science has I don't know why we are keen for these difficult names.
Latin names.
Plexus. Why it comes from? What are these plexus? Is like just a group of nerves, or
That's
no?
a whole. I think it's a whole layer of neurons.
Oh, whole neuron.
And
So both answers were correct, actually.
yeah.
Good. So this was a bonus question, I suppose. So everybody got it right. Okay.
Yeah.
Next one. So how much serotonin is produced by the second brain? Ooh, I think I know. I think, but again, tricky, very close values. Oh, but most people actually got it right, 95%. I think the association with the chocolate was a good one.
And at the end of the talk, so they still know it.
Ooh, I like some names.
Yeah.
There was one hungry for chocolate.
Last question. So what is the scientific name of the second brain? Is it autonomic nervous system? Is it sympathetic nervous system? Is it enteric nervous system? Or is it parasympathetic nervous system?
Well,
so know your names.
Well, almost everybody got it right. Everybody paid a lot of attention, because this one, enteric nervous system. Well done. I'm curious to see who is on top. So third, X. Second, Almeida, and first.
We have a winner.
Nev.
Nev.
Congrats.
Cheers to Nev.
Do we have
Nev,
contact details of Nev so we can send them fast, or how shall we contact this winner?
Indeed. Maybe the winner can contact us directly either via our social. Indeed, you can just send an email. That's a good one. With Nathalie. Okay, that's congratulations, Nathalie.
Yeah, so you can send us an email with your contact details, so we'll make sure you'll receive the pint glass. So congratulations.
Very nice. Okay, I was wondering, while people can still ask some final questions. In the meantime, I think you, Linda, you had already one or so already.
Yeah, so I also had a question about the role of the microbiome, but I think you already mentioned that you are not really focusing on that. So I was just wondering about your opinion about these probiotics, whether they really impact the second brain, and whether a microbiome transplant, for example, would help in like maybe avoiding a lot of diseases if your microbiome, but maybe you're not.
Yeah, I can answer that, I guess. Like there is like, so we do also a little bit of microbiome because you cannot neglect it totally if you're studying the second brain. And yeah, as you mentioned in the beginning, I also work in Rotterdam, University of Rotterdam, and there currently I'm doing together with a postdoc, Laura Kuil, we're doing a systematic review on investigating how these prebiotics work in the context of if they are helpful. So there are many studies. I think they're also very contradictable studies. I think it's also what I heard is hard to compare because the researchers used many different prebiotics or probiotics. So I don't know. I don't know the outcome. If it's really helps, that I don't know, but I will tell you once we have done this systematic review, and we do it in the context of obesity, by the way. That's one. And then the second, I'm a believer of these fecal transplantations. So I think there's future in it, but I think it's too early to say because as you know, there's so many bacteria in our gut, and I think it's very hard. There are many assumptions, but I'm not totally in the literature, but what I have the feeling, they're often assumptions and like very hard facts, and to compare different studies and to really come with a decent conclusions. I don't know, at least I know it's out there for many diseases yet, but probably it's future.
Yeah, and you also see that the microbiome can already differ between different mouse models that you use. So if you have a mouse model that doesn't have an immune system or something, they can have a different microbiome and stuff as well. And that has different effect, or at least that's what they think, that it has a different effect on the second brain, because the structure of the second brain is then a little different or altered as compared to what you can see in like a normal mouse model.
Yeah, and is it really easy to control the composition of your microbiome, like of mice, like siblings, or I don't know? Is that easy, or because I have no idea?
I know you really need like high numbers if you would like to compare things, and probably if you keep them in the same environment and if they're genetically the same, that helps. I'm really not sure, but with a human, there's such a huge difference between all humans, so therefore I think it's hard to make like really conclusion, but I think it's really a field where we hear from more in the future regarding treatments and medication and everything.
Yeah, it's a very interesting field indeed. I think Margarita had also some questions left, or you want to ask them?
Well, I was wondering, I don't know, if so we heard a lot about communication, like this second brain. I was wondering if our gut also communicates with other, I guess it communicates with other organs. I don't know, like if it, because here it's more the communication between the brain, I guess, sometimes. Yeah, the brain and our second brain. But if also besides this big communication, if it's known that the gut also communicates with other type of organs, and which situation?
I think it's a really good question. I don't know particularly the answer. It's they say the enteric nervous system is part of the peripheral nervous system, and if you would look at the body that we showed you, the cross section, they're all interconnected. So you would indeed assume that they also talk to each other. Now people only look communication brain gut, and gut brain, and gut other organs, I think. But I don't know. I think it's very interesting. I don't know if the second brain is a primary regulator of other organs. I don't know. As far as I know, there are also no studies. So interesting question, interesting new field probably that you propose.
Yeah, maybe all about communication. Because I was wondering, imagine like when we drink coffee, visually you feel like going to the toilet a few minutes afterwards. And I don't know, I always thought was maybe the coffee like hitting the brain and then communicating to the guts, but probably it doesn't even need that. probably, I guess it's so. What's is it known what happens during this process?
So I think it's known that when you have to go to the toilet, at least it's your first brain really makes a decision. Yeah. You go or you don't go. Your second brain is not conscious, or how do you call it? So they cannot. I don't think your second brain doesn't decide. So it's your first brain. So your second brain controls everything from the movement till your palate, your features at the end of your anus, and then to say go or no go, that's your brain who's connected there towards your anus, who tells you like, okay, now you can go. And that's lucky us because
at least have the time to go.
Good. Thank you for your answer. I think there's no more questions. I think it was a great talk. I really enjoyed it.
Thank you.
And
thanks a lot.
Yeah, very cool fields. A lot of cool directions.
There's still
a lot of music.
Yeah, so I guess quite in the early phase of right. Still a lot to be done.
Yeah, it's really booming now. I think we will know much more in the upcoming years, because like first it was a really small field. There were only like 200 researchers really focusing on it, like a small field. But now it's booming. I think it's everywhere. People, macrophages, interaction with second brain, first brain. Yeah, it's everywhere. So I hope and I think there will be a lot of new knowledge in the upcoming years. Yes.
Yeah, it would be a very interesting field indeed to maybe prevent a lot of diseases from occurring. That would be great, of course. If you could start with the health of your second brain, then that would be really nice. Yes, indeed.
Yes.
So I think we need to wrap up the event. There is more events next week, so I really recommend you to tune in again next week, because we have some other interesting events also planned by the other cities. And I think, we would like to thank the speakers and our audience, of course, for their nice questions. And wish you all a really nice evening and a really nice Ascension weekend break for the ones who have that.
Yes. Maastricht we have it.
Yes. Okay, so cheers.
Cheers. Thank you very much for having us. Bye-bye.
Bye.
Bye.
Bye. Cheers.